Chapter 18, Infectious Diseases Affecting the Skin. First, we'll look at the anatomy and normal defenses of the skin, things that keep the skin from getting infected. So the skin is made up of several layers. The most superficial layer is called the epidermis, and the top portion of the epidermis is the stratum corneum, which is comprised of dead skin cells that are about 40 cells thick, and the entire epidermis is replaced every 25 or so days. Underneath the stratum corneum, we have the basal layer, which is made up of epithelial cells. And then in the deeper tissues, the dermis, we have all of the blood vessels, the hair follicle shaft, the sebaceous gland, which secretes oil. We have the nerve fibers. And then below the dermis is the subcutaneous layer comprised mostly of fat. If you injure the epidermis, it does not result in bleeding, but injury to the dermis or the subcutaneous fat layer would result in bleeding from the wound. The protections of the skin are multifold. So first of all, the stratum corneum, which is comprised of dead skin cells, is a very dry environment, so it's difficult for microorganisms to grow in the absence of water. We also have sweat, which is released and contains lysozyme, an enzyme that will degrade the bacterial cell wall. Sweat also contains salt, and it has an acidic pH, both of which will prevent growth of microorganisms. We have the sebaceous gland, which will release oil that has a slightly acidic pH and will reduce growth of microorganisms on the skin. And then we have the epidermis being made of very tightly packed skin cells, which release a lot of protein called keratin. So this layer is said to be keratinized, which means that it is very waterproof and it's a very durable, continuous structure. So it's difficult for microorganisms to penetrate and get into the host in the deeper tissues. And then lastly, we have the surface is constantly being sloughed off, so we're shedding dead skin cells continuously. So even if a microorganism gets established in the low water, low pH, and high salt environment of the skin, it's likely that it will be just shed off as the skin layer is shed. And then we also have the presence of normal biota. So we found in the Human Microbiome Project, which was trying to categorize and find numbers of microorganisms living in and on humans, there are quite a lot of normal biota in a healthy human. So different types of bacteria, there's also yeasts, I'm sure there's a lot of viruses as well. So all of these normal biota will additionally compete with pathogenic microbes for nutrients and growth in this niche. And all of these normal biota that grow on the skin in a healthy person are very well adapted to the salt and pH and dryness of the skin environment. Okay, so the first of the infectious diseases of the skin that we'll discuss is methicillin-resistant Staphylococcus aureus, or MRSA. So methicillin is a type of antibiotic, and Staphylococcus aureus have acquired resistance to this antibiotic. They've also acquired resistance to several other types of antibiotics, making MRSA a very difficult infection to deal with. So we'll have these disease charts for each of the diseases, which list the main factors and the summary of the disease. So what we're going to focus on with MRSA is the virulence factors, because Staph aureus has quite a lot of virulence factors. So we have coagulase, which is an enzyme made by Staph aureus that causes blood clotting. And this is a way for Staph aureus to hide in the host. If it can cause a blood clot to form around it, then it can evade immune defenses. Staph aureus also produces hyaluronidase, another enzyme which is capable of degrading connections between epithelial cells and the superficial layers of the skin. So when Staph aureus releases hyaluronidase, it will then break apart connections between skin cells, enabling the Staph aureus to migrate through the host and get into deeper tissues and spread the infection. We have Staph aureus can make staphylokinase, which is another enzyme capable of breaking apart host tissues, enabling Staph aureus to spread. DNase is an enzyme that can degrade nucleic acid, so in this way it can damage host cells by breaking apart their DNA. And then lipase is an enzyme that some Staph aureus can make, which will break down oils, which are secreted by the sebaceous gland in the skin, and the production of lipase will help Staph aureus to survive in the skin environment. Staph aureus in the top row listed as also being incredibly resistant not only to antibiotics but also to salt, pH, temperature, and many disinfectants. So it is an incredibly persistent and hard to kill infection. It can infect both non-hospitalized people, typically will stick to the skin, but it is also a healthcare associated infection, can be found in patients in the hospital, and it's often a blood infection in these individuals. So we'll talk now, we'll first look at a picture of MRSA, and then we'll discuss diagnosing it, because there are several ways to diagnose this infection. First, we see the appearance of MRSA, so it's often a raised sore. It's very hot to the touch and very red around the area, and then there's a lot of pus filling up the lesion. So one of the first things that will be done with a MRSA infection is to drain the pus and try to release some of the pressure and the soreness. Diagnosing MRSA is usually done by taking a swab from the wound and then looking at it under a microscope. MRSA is a gram-positive staphylococcus, so it's going to be rounded in shape, and its arrangement will be grape-like clusters for the staphylo. If the wound contains a lot of different kinds of microbes, you can grow MRSA on mannitol salt agar, which is shown in B. So mannitol salt agar is a petri dish which has an agar substrate for the bacteria to grow on. The agar substrate is pink, contains a lot of salt, and it also contains the carbohydrate mannitol, which will serve as the energy source for the bacteria. So mannitol salt agar is selective for Staphylococcus. So Staphylococcus is very resistant to salt and so can grow on mannitol salt agar, but many other microorganisms cannot grow on this type of agar. So that's why we say it's selective for Staphylococcus, because only Staphylococcus will grow on mannitol salt agar. And when the Staphylococcus grows on mannitol salt agar, it will use up the mannitol carbohydrate and it will break it down, metabolize it, and this will cause production of acid, which will change the media from pink to yellow. So you can see yellow areas where Staphylococcus is growing on mannitol salt agar. Okay, in C, there's another diagnostic test. So from the patient swab, we can add that to a test tube containing plasma, which is the liquid part of blood. So this is often isolated from rabbits or goats. So you would add the patient swab with the plasma into a test tube. If the microbe produces coagulase, which is an enzyme that causes blood clotting, and Staph aureus often does produce coagulase, the coagulase will turn the liquid plasma into a solid. So if you incubate it or leave the swab with the liquid plasma overnight, the next day, if the liquid plasma has turned solid, then you can conclude that there was a microbe in the swab making coagulase, which caused the liquid to clot. So in this figure, the bottom two tubes, we have the bottom two are both positive because they're turning the liquid plasma either partially solid or completely solid. And then last, you can take the patient swab and you can spread it onto a blood agar plate, which is a petri dish containing an agar substrate that has blood added to it, so it makes the agar very red. And Staph aureus will make enzymes that will lyse the blood in the agar and cause the red to clear into a completely clear zone around where the Staph aureus is growing. So this is a little hard to see on part D, but if you grow Staph aureus on blood agar, you'll see an area where the red is made clear by production of enzymes. Okay, second skin disease is impetigo. So this is usually caused by a mixture of Staph aureus and Streptococcus pyogenes, another gram-positive bacteria. It's really common in preschool-aged children. And the virulence factors for these microorganisms are that they make toxins called exfoliative toxins, which are going to break down the structure of the skin and cause a really specific looking rash, which I'll show you in a second. And these bacteria have received the gene information for making exfoliative toxin by bacteriophage. So this is an example of transduction, where the bacteriophage virus gives new genes to the bacteria. In impetigo, we can also see production of coagulase, which is the enzyme that clots the liquid part of blood. And then the bacteria can be coated in M protein and or a capsule, which will both help with host adhesion, so adhering to host tissues, and also prevention of phagocytosis. Okay, so let's look at a picture of impetigo, and then we'll talk just a little bit about diagnosing it. So the impetigo rash is often on the face, and it's often in preschool-aged children. The rash is described as being honey-colored and flaky, so it kind of looks like a really dry area. And the reason the rash looks this way is because of the production of exfoliative toxins by the bacteria, which cause the layers of the skin to separate and start flaking off. Diagnosing impetigo can be done by visual inspection, so just looking for the honey-colored flaky rash. Also, a catalase test can help distinguish if the impetigo is caused by Staph aureus or Streptococcus pyogenes, because the Staph aureus is the only bacteria that makes catalase. And a catalase test is shown on the left. So what you do in this test is you take a sample from the wound, you would spread it on a glass slide, add a little bit of hydrogen peroxide, and if the hydrogen peroxide bubbles, then the microbe is turning the hydrogen peroxide into water and oxygen, which causes the bubbling. So in this experiment, we have a microbe that produces catalase on the top, so you can see the bubbling, and on the bottom, we have a microbe that does not produce catalase. So catalase is indicative of Staphylococcus aureus. Also, spreading a swab from the wound onto blood agar, and then seeing if the microorganism is sensitive to bacitracin, which is an antibiotic. So the way this is done is you spread the microbe on blood agar, and then you put a paper disc, which is marked A in this figure, that has been soaked in the antibiotic, so in this case bacitracin. You put the paper disc on top of the agar where you've just spread the microbe. The antibiotic will seep into the agar, and there will be an area around the antibiotic-soaked disc where there's no growth of the microorganism, which indicates the microbe is killed by the antibiotic bacitracin. So in this result, we see an area around the disc marked A that's been soaked in bacitracin, where the microorganism is not growing. It's a little bit darker in color. And so this tells us that the microbe is killed by the bacitracin, and therefore the microbe is Streptococcus pyogenes. We also see a characteristic growth pattern called beta hemolysis for Strep pyogenes, which can help diagnose it as well. Okay, the last of the skin diseases is human herpes virus 3. So this is a viral disease which is a member of the herpes family, and there are several of these, including the sexually transmitted infection herpes. This one is the virus that causes chickenpox and shingles. It's also called varicella virus or varicella zoster virus. It is an enveloped virus, so it has that extra lipid layer outside of its capsid, and it has a DNA genome. Transmission of human herpes virus 3 is by droplet transmission or direct contact with active skin lesions, and there's almost a 100% infection rate with exposure. The main virulence factor of this virus is that it can remain latent in dorsal root ganglion. So when a virus goes latent, it infects the person, makes them ill, and then goes through a period of inactivity where the person is outwardly healthy, but the virus is still in the host. And at a later date, the virus can be reactivated and cause an illness again. So human herpes virus can infect a person, cause chickenpox on the first infection, then will go latent by getting into nerve cells along the spine called the dorsal root ganglion. The virus stays in these nerve cells until later reactivation, where the virus reemerges to cause shingles. The incubation period is long for this virus, and it results in a rash over the entire body. And these rashes is a pox rash, which means it's a pimple-like sore that's filled with fluid and can cause pock marks after healing. In very rare cases, it can cause encephalopathy, which is swelling of the brain. It can be a really serious infection in newborns, or if a mother is infected and the fetus gets infected, it can cause birth defects. Diagnosis is just appearance of the lesion, so the way they look can indicate that you have chickenpox or shingles. And the virus is self-limiting, meaning that it will eventually clear. You just have to wait for the disease course to progress. There is a prevention vaccine for infants, which can prevent chickenpox, and then it's recommended that individuals age 65 and over get a shingles prevention vaccine just to make sure you don't get shingles. So this is what the pox look like. They cover the entire body on the first infection when you have chickenpox. And then the virus can go latent in these nerve cells surrounding the spinal cord. So after the chickenpox infection clears, the virus will go into these nerve cells and remain latent for years or decades, and then will reemerge as shingles, which always has a specific pattern, normally on the trunk where these dorsal root ganglion cells are, and it will always be on one side and stop abruptly at the midline of the body. So that concludes chapter 18.